Okay, this is Dr. Morris. I'm continuing the lecture series with Chapter 18, which deals with the practical applications of immunology. We'll start with talking about vaccines and the principal effects of vaccination. A vaccine basically is a suspension of microorganisms or some part of the microorganism that will induce immunity when it's administered to the host. And recall, the host is the human subject. Vaccines had their origin from the practice of variolation, in which material from smallpox scabs were basically inoculated into the bloodstream to provide immunity against the disease. We now call this strategy vaccination or immunization. A disease can be controlled if, but not all, of the population is immune. So if all of the population is immune, is referred to as herd immunity. Now again, most of the population, I guess I should rephrase that, is controlled if most of the population is indeed immune to the organism or the infection. It doesn't have to be all of the population that's affected. Microorganisms used in vaccines may be inactivated or killed, the whole agent or an attenuated agent of the whole agent. So anything that weakens the microorganism so it cannot cause an infection is referred to as attenuated or weakened. Attenuating vaccines tend to mimic the actual infection and usually provides better immunity. A subunit vaccine uses only those antigenic fragments of a microorganism that are best suited to stimulate the immune response. These subunits may be produced by bacteria or yeast, for example, may use genetic engineering techniques such as vaccines are called recombinant vaccines, and we talked about this earlier in the semester. Now, toxoids are inactivated bacterial toxins. If I may regress for a moment and talk a bit about the subunit vaccines. So remember, sometimes it only takes a small part of a particular agent to initiate the immune response from the body. And if that small fragment is a part of the microorganism, typically that's enough to initiate immunity. So it can just be the cell wall of a particular microorganism. It may be an inner structure, or again, as referred to, an antigenic fragment, any part of that microorganism that can stimulate that immune response. Vaccines can be fragmented and desired antigens separated out. Rather than using a complete cell, these are called acellular vaccines, and some polysaccharide vaccines have enhanced effectiveness when combined with tetanus or diphtheria toxoids. These are called conjugated vaccines. So again, when we look at vaccine and its history, virilization, this is the inoculation of smallpox into the skin. This was done in the 18th century. Vaccination, or the inoculation of cowpox into the skin. The term herd immunity refers to or results when most of the population is immune to a disease. Now remember, that's most of the population, not necessarily all of the population. Some principal vaccines used in the United States to prevent bacterial disease include diphtheria, pertussins, and tetanus. We also have vaccines for meningococcal meningitis, Haemophilus influenza, and forms of pneumococcal. Some other principal vaccines used in the United States, or that have been used in the United States, would include smallpox, which is the live vaccinia virus. Vaccinia refers to the agent that causes cowpox, but again, the similarity between cowpox and smallpox is such that if you administer the vaccinia virus, it will also confer immunity to smallpox as well. Poliomyelitis, which is an inactivated virus. Rabies, also an inactivated virus. Hepatitis A vaccine, inactivated virus. Influenza, inactivated or attenuated. Attenuated, remember, means weakened. Measles, the attenuated virus. Mumps, the attenuated virus. Rubella, chickenpox, both attenuated viruses. Then hepatitis B, which is an attenuated, or I'm sorry, is a antigenic fragment, a recombinant vaccine. The term adjuvants refers to chemicals or an alum, for example, added to a vaccine. It improves its effectiveness. Nucleic acid vaccines or DNA vaccines, these are naked DNA into muscle. The result is the production of proteins encoded as a result of DNA, which persist and stimulate an immune response. RNA, which could replicate in the recipient, might be a more effective agent. Diagnostic immunology. Basically, we cannot see antibodies and must infer their presence in a variety of reactions. For example, in a precipitation reaction, this involves the reaction of a soluble antigen with the immunoglobulins IgG and IgM antibodies to form large interlocking aggregates called lattices. Precipitation can be easily arranged by allowing the antigen and antibody to diffuse toward one another. A cloudy line of precipitation forms in an area which its optimal ratio has been reached is called the zone of equivalence. In a capillary tube, this is called a precipitin ring test. In a petri dish, the antigens and antibodies may be placed into wells cut into an agar medium. This is called an immunodiffusion test. If passive diffusion of reactant is too slow, the test can be speeded up by amplifying or applying an electrical current. To the procedure is referred to as electrophoresis. Immunoelectrophoresis is commonly used in many studies. It's used in research to separate proteins in human serum. The precipitation reaction, again, is seen here. Soluble antigens, you see the zone of equivalence, and then you see the antibodies, which are separated out. This is it in the actual slide. It involves soluble antigens with antibodies. A glutination reaction involves particulate antigens that can be linked together by antibodies in a process referred to as agglutination. Direct agglutination tests detect antibodies against relatively large cellular antigens such as red blood cells, bacteria, and fungi. The amount of antigen in each well of a microliter plate is the same. The serum containing the antibodies is sequentially diluted out in a series of wells. The higher the concentration of antibodies in the serum, the more dilution would be required to dilute it to a point at which no reaction occurs with the antigen. This is a measure of the titer, or the concentration of the antibody. Now, for diagnostic purposes, a rise in the titer during the course of a disease indicates that antibodies are associated with the disease. If it can be shown that a person's blood has no antibody titers before an illness, but develops one while the disease is progressing, this is referred to as seroconversion and is diagnostic. You should be familiar with the definition of seroconversion. Soluble antigens can respond to a agglutination test if the antigens are adsorbed onto particles, especially in minute latex spheres. In such indirect passive agglutination tests, usually called the latex agglutination test, the antibody reacts with soluble antigens adhering to the latex particles. This is the basis for many new diagnostic tests. The illustration here from your textbook demonstrates the agglutination reaction. It involves particulate antigens and antibodies. Antigens may be on a cell, or direct agglutination, or attached to latex spheres, which is indirect and or passive agglutination. Now, hemagglutination. Certain viruses have the ability to agglutinate red blood cells. The process is referred to or called viral hemagglutination. This is not an antibody-antigen reaction. Hemagglutination involves agglutination of red blood cells, whereas viral hemagglutination inhibition tests for antibodies by the antibody's ability to prevent the viruses from agglutinating to red blood cells. As you can see here, the viral particles are stuck between the red blood cells, thus the ability to prevent the virus from agglutinating red blood cells. The harmful effect of bacterial exotoxins or a virus can be neutralized by specific antibodies. One such substance is called an antitoxin. This is used in the treatment of diphtheria. Antitoxins produced by other animals can also be injected to provide passive immunity against toxins. Similarly, if an antibody binds a virus, it prevents it from attaching to a host cell and infecting it. Again, the textbook shows you the harmful effects of a virus or an exotoxin. Antiviral antibodies in the serum and a virus in the serum. The virus neutralized and the hemagglutinin is inhibited. Viral hemagglutinin tests detect antibodies to a virus. These viruses will normally cause hemagglutination when mixed with red blood cells. If the antibodies to the virus are present, as is shown in the illustration above, they neutralize or inhibit hemagglutination. If a person's serum contains antibodies against the virus, may cause hemagglutination, these antibodies will react with the virus and neutralize them. This is the basis of viral hemagglutination inhibition tests. It's used in the diagnosis of influenza, measles, and in mumps. You should be familiar with the types of diseases where you see the viral hemagglutination test. Again, influenza, measles, and mumps. Now, there's also a reaction referred to as the complement fixation reaction. Complement is a group of serum proteins that we talked about in the previous lecture. During most antigen-antibody reactions, the complements bind to the antigen-antibody complex and is fixed. This complement fixation is used to detect the presence of antibodies. Antibodies that do not produce a visible reaction by other means can sometimes be demonstrated by fixing complement during the antigen-antibody reaction. Another study is referred to as the fluorescent antibody technique. The fluorescent antibody techniques use fluorescent dyes or fluorescein that is combined with the antibodies to make them fluorescent when exposed to ultraviolet light. This requires a fluorescent microscope or fluorescent microscopy with ultraviolet light illumination. If a direct fluorescent antibody test, the antigen can be fixed onto a slide. A test serum is known upon the antibody is added. If the serum contains antibodies against the test antigen, it will bind to it, but this is not visible. To visualize the antigen-antibody complex, fluorescein labeled anti-human gamma globulin, an antibody that reacts specifically with any human antibody, is added to the slide. Unbound reagents are washed off the slide, which is examined under a fluorescein microscope. If the known antigen on the slide appears fluorescent, antibody specific to it was in the test serum. A fluorescent activated cell sorter uses the fluorescent antibody markers to isolate and identify the cells, such as the T cells. One of the most widely used serological tests is the enzyme-linked immunosorbent assay, referred to as ELISA. This is the most widely used of the group of tests called enzymed immunoassays, or EIA. A microtiter plate with numerous shadow wells is used into two basic methods that we will describe. A variation of the test may be used or may use reagents bound to tiny latex particles rather than the surface of the microtiter plates. In the direct ELISA, the goal is to identify an unknown antigen such as a drug in a serum sample. In the illustration that is identified here, the enzyme-linked immunoassay, or the direct ELISA, you see the antibody is absorbed into the well. The patient sample is added, complementary antigens will then bind to the antibody. The enzyme-linked antibody specific for the test antigen is then added to the antigen, forming a sandwich, illustrated here. The enzyme substrate is added. The reaction produces a product that causes a visible color change. In the indirect ELISA, the object is to determine the presence of certain antibodies in the serum. The assay currently used to test antibodies against that in the AIDS virus. In both these variations of the ELISA, a positive reaction is indicated by a color change in the well due to the action of an enzyme linked to the last antibody added and allowed to react with substrates added in the final step. This is the indirect ELISA. We see antigens are adsorbed to the well. Patient antiserum is added to the complementary antibody and binds to the antigen. The enzyme linked is added and binds to bound antibody. Enzyme substrate is then added and the reaction produces a product that causes a visible color change. There are serological testing that's done as well. In serological tests, we have direct tests which detect antigens from a patient sample. We have an indirect test which detect antibodies in a patient's serum. Just as a review, we've talked about agglutination, particulate antigens. We've talked about hemagglutination, which is agglutination of red blood cells. We've talked about precipitation of soluble antigens. We've talked about the fluorescent antibody techniques or antibodies linked to fluorescent dyes. Complement fixation, which have red blood cell indicators. Neutralization, which inactivates toxins or viruses. And then the ELISA, or the peroxidase enzyme, is the